Hello, everyone, and welcome to our Pharmaceuticals webinar, first one this year, in the topic of recent advances in ocular pharmacology. My name is Tatiana, and I will be your host for today. So I will share a bit about MDPI. MDPI is a pioneer in scholarly open access publishing and has supported academic communities since 1996. It is based in Basel, Switzerland, and has the mission to foster open access scientific exchange in all forms across all disciplines. We are now up to 472 diverse and open access journals, 463 of which are peer-reviewed and 9 are conference journals. And they are supported by more than 295,000 academic experts who share our mission, our values, and our commitment to providing high-quality services for our authors. When it comes to pharmaceuticals, it is one of our international open access scientific journals of medicinal chemistry and related drug science. This journal welcomes manuscript covering a wide range of aspects involved in drug discovery and development. And mostly we are publishing next topics. Those are small molecules and drug candidates, biomolecules, natural products, biological targets and biomarkers, radiopharmaceutical science, pharmacokinetics and pharmacodynamics, and pharmaceutical preparations and drug delivery. Today's program looks like this. We might need to make some adjustments. However, we are looking forward to hearing our 4 speakers. It should be great. And in the end, we will have Q&A sessions. So please, if you have any questions, feel free to leave them in Q&A box in the bottom. So you can name who the question is for, and we will answer it in the end. We also have a relevant special issue connected to this webinar. It has the same title, Recent Advances in Ocular Pharmacology, and guest editors are the chairs of this event. The deadline for a manuscript submission is the end of July. So if you have something to submit to this special issue, please feel free to do it until then. So as I mentioned, there is a Q&A box. Feel free to leave all of your questions in it, and we will make sure to answer it in the end of this event. I will now share who our chairs are. Our first chair is Professor Ioannis Tsinopoulis. I hope I pronounce it well, from Aristotle University of Thessaloniki, and he is Associate Professor in Faculty of Medicine, Department of Ophthalmology. He does his research in ocular cell cultivation and nanotechnology application in ophthalmology. He has quite a big clinical research and is part of several committees in his university. Our second chair and second guest editor of the special issue is Dr. Lampros. I have really big issues with your names. Coming from Ophthalmica Institute, also in Greece. And he is specialized in pediatric ophthalmology and strabismus. He is working on nanotechnology and cutting-edge technologies in pediatric ophthalmologies. So without further ado, I will share the floor with our chairs, and they will announce our speakers.
Okay. Thank you very much, Tatiana, for this invitation. I am Dr. Professor Tsinopoulos, and okay, dear all, I am very happy to welcome you to the pharmaceutical webinars webinar regarding recent advances in ocular pharmacology. I want to thank you, our guests, who will kindly share their expertise on this interesting topic. And I also wish to thank all the members of the pharmaceutical team and you, Tatiana, who invited us to edit this special issue and have done their best to support us. At this point, I would like to publicly congratulate Professor Dimitris Fatouros, συγχαρητήρια Dimitri, on his selection as president of the Department of Pharmacology at Aristotle University of Thessaloniki. I will now ask my colleague and my good partner, Dr. Lambros Laprogiannis, to take over the introduction. Thank you.
Thank you very much, Professor Tsinopoulos. Thank you very much, Tatiana, for your kind and polite introduction. I would like also on my behalf to welcome you all to this webinar, both the presenters and the scientists who are devoting this difficult part of their day to listen to us, to our presentations. I'd like to particularly thank the presenters, and it's a great honor that we have the opportunity to present, to listen to them presenting their work in the field of ocular pharmacology. And this is a particularly interesting topic, as we understand. There are not only new substances, but also treatment modalities and delivery pathways that are being discovered literally every day. And this is the presentations that we will be lucky enough to listen to today are on the edge of this progress. A great part of this webinar will be devoted to the ocular barriers, which is a very big part of what makes ocular pharmacology a topic in itself. And I think that without further ado, I would like to present you the first speaker, Professor of Pharmacology Antonios Voulas, who is sharing my name in his screen, unfortunately, but I'm certain that this can easily be fixed. Thank you very much for joining us today, Professor Voulas. I'd like to briefly say about your work that you are a tenured professor with the first laboratory of pharmacology in medical school and with the Aristotle University of Thessaloniki. And you have an extensive record of training, teaching, and participating in publications, books, and journals concerning pharmacology. And we would be more than happy to listen to your presentation regarding the ocular barriers. I'd like to give you the floor, please.
Yeah, thank you very much, Lambros. Thank you very much, Tatiana, for the kind invitation. Well, I guess, yes, my presentation is going to be mostly introductory, not much recent research, rather an overview of what is known regarding the peculiarities of pharmacokinetics of the eye. So let me share it with you. All right, so as I said before, mine is an introductory presentation on the particulars of ocular pharmacokinetics, which, by analogy to pharmacokinetics in general at organismal level, it describes the processes of absorption, distribution, metabolism, and elimination of drugs in and from the eye. Now, what makes the eye different from the organism as a whole or from the more amply perfused organs is that it is relatively isolated from its environment and from systemic circulation through either static, in other words, anatomical, such as the cornea or the retina, and dynamic or physiological barriers, such as, for instance, the nasolacrimal drainage or the blood flow or the lymph flow and so on and so forth. Which means that the bioavailability of ocular drugs depends largely on their ability to cross static barriers and to resist the action of dynamic ones. Yeah. So let's see what these barriers are. As I said, we have the static and the dynamic barriers. The static barriers include the blood-ocular barrier, which again includes in turn blood-aqueous barrier and the blood-retinal barrier. Okay, the blood-aqueous barrier is mainly located here over in the ciliary body and the iris, and it consists of epithelial cells that are tightly are in tight junction with each other. They form tight junctions with each other, so not much can pass through them by itself. The exception being if there are transport proteins that help channel things, back and forth. And of course, there's the endothelial barrier, which can be more or less tight also in turn. And in some places, it's less, it's more conductive than in others. And in the retina, we do have the blood-retinal barrier, and here also, this consists of the epithelial barrier, which normally is more tight, and the endothelial barrier, which can be at times fenestrated, so more things can go back and forth. But in total, this is a quite tight barrier. It's not as tight as the blood-brain barrier, but is very tight, I would think. Now, as I said before, there are transport proteins. There are proteins that help channel things into the eye, but there are also efflux pumps, which they tend to recognize mainly lipophilic molecules, lipophilic drugs, and they push them back out. In the posterior part of the eye, we do have the corneal and the conjunctival barrier. Corneal barrier, of course, is more exclusive. And this is a quite complex barrier because it consists of different layers. Some are more lipophilic, some others are more hydrophilic. And as we're going to see, this puts a strain into the physicochemical properties that the drug has to have in order to be able to cross the corneal barrier. Now, the routes of administration of ocular drugs, as we know, are mostly topical. Okay, so ocular drug delivery is usually topical on the outer corneal surface. But other common routes, or relatively common routes, include the intravitreal route, the subconjunctival route, and less common, less usual routes are the retrobulbar, the peribulbar, the subtenon, and the intracameral routes. Now, once a drug molecule or a formulation containing a drug molecule, to be more precise, is applied on the outer surface of the cornea of the eye, okay, it has, as I said before, it has to go through a number of different layers. which have different properties in themselves, in order to finally get into the posterior chamber, the anterior chamber, sorry. Now, the first of these layers is the tear fluid, which is covered by an oil layer. And this puts a requirement for lipophilicity to the drug, to the molecule that has to go through it. On the other hand, once the molecule has gone through the mucin layer, which is another, let's say, restrictive layer, because some compounds tend to get attached to it more than others. Now, once the drug has gone through those layers, it enters the stroma, and the stroma being, of course, a very hydrophilic milieu, turns the table around, and now the drug has to rely on its hydrophilicity in order to be able to go through and get to the other side. So, as I said before, this is a quite complex situation. Now, obviously, because of this complexity, the intracorneal penetration is the rate-limiting step in penetration into the of the drug into the anterior chamber. Now, as we can see here, the graphic on the right-hand side, the rate of absorption usually follows first-order kinetics, as is the case for most drugs when they are absorbed also from the intestine. Okay, which means that the more drug you have on the outside, the faster the drug will go into the inside of the eye. In other words, this is a concentration-dependent process. Now, that means that drug concentration at the outer surface of the cornea is probably the most important factor for the rate of absorption. But drug concentration is also limited by a variety of factors, and here comes the dynamic barrier that we talked about at the beginning, such as the nasolacrimal drainage and also conjunctival absorption. Now, both these two processes tend to channel the drug away from the cornea, so the drug doesn't go in. Once it's carried over by the tear and into the following the nasolacrimal drainage process, or if it gets absorbed into the conjunctiva, because the conjunctiva has a lot of capillaries, and the drug will easily go into the systemic circulation from there, and will, of course, miss the eye. Which means that to the extent that a large proportion of the drug, instead of going into the eye, ends up in the systemic circulation, that creates a big potential for adverse effects. And this is something that, unfortunately, we tend to forget when we think about topical application administration of drugs in the eye, because when we study pharmacology, eye is not usually the organ that we have in mind, and we tend to, we think that everything that's applied topically on the organism misses the systemic circulation. Now, as I said before, a large part of the formulation will tend to miss the eye. It will never end up in the anterior chamber, and which means that the percentage of the dose which is usually absorbed is somewhere between 5 and 10%. Okay, and out of this 5 to 10%, also a small percentage will end up in the conjunctival and scleral roots, rather than go through the cornea and penetrate the cornea and end up in the anterior chamber, which means that the small percentage overall, okay, will end up where we want it to end up. Now, as for the rest, as I said before, most of it will end up in the systemic circulation because all these epithelia that we see in the in this box in the right-hand side are they have a thick network of capillaries, and it's easy for the drug to get absorbed through them. Now, what does this mean in practical terms? It means that aqueous humor bioavailability, which means the extent, the percentage of the drug that ends up unchanged, intact into the aqueous humor when the drug is applied to the exterior of the cornea, increases with formulation volume, but remains constant over 20 microliters. So no matter how much of topical formulation one applies onto the cornea, the rate of absorption will not increase, and the bioavailability, of the drug into the aqueous humor will not increase over 20 microliters of initial volume. Now, there are some factors that can increase bioavailability, and these are taken advantage of by people who design ocular formulation, as Professor Fatouros is going to tell us in the next presentation. And these factors include viscosity, the highest the better, pH, there's an optimal pH of 7 to 7.7, lipophilicity, which has to be intermediate, and size, meaning that as in general pharmacokinetics, general absorption consideration, means that the molecule has to have a rather small volume, because otherwise it will never go through the epithelial and the endothelial barriers. Now, in our days, there are formulations that include various excipients which increase the viscosity, but not just that. They provide a matrix which has, it can have mucus adhesive, for instance, or penetrating properties, or it has a charge density which allows it to interact with various eye components and thereby increase its presence into the various eye tissues. And we can see some examples in the right-hand side graphs here, especially with regards to the aqueous humor. We can see a big increase, okay, large increase in bioavailability of this particular drug once it is combined with what is called here a mucus penetrating particle. Also another example here which shows us that the Cmax, the maximum concentration into the aqueous humor, can go up to 20 times or even higher once the formulation contains a particular compound which helps increases the rate of absorption. And we also have impressive increases with regards to the area under the curve, and a very dramatic decrease with regards to the time that's needed for the drug to reach the maximum concentration into the aqueous humor. So there's a lot of interesting research in this topic. Now, once the drug is into the ocular tissues, okay, it can interact with various factors. And one of those factors, a very important factor, is the melanin molecules that are situated within the iris and the ciliary body. And well, this interaction is like a double-edged sword for the pharmacokinetics of the drug, meaning that on one hand, they can accelerate aqueous humor clearance, meaning that the drug is removed from the aqueous humor because it is tightly bound into the iris or the ciliary body. On the other hand, this binding is reversible, meaning that this process can serve to produce a sustained release effect. And this can be a good thing. And what on our right-hand side of the slide, we can see a very impressive demonstration of this effect, this process. As we can see, the change in concentration of the drug, which here is a beta blocker, bifunolol, follows the drop in concentration in the cornea and the aqueous humor. Okay, in the case of the albino rabbit, where there is no melanin, of course, molecules in these tissues. Whereas in the pigmented rabbit, this concentration, the corresponding concentrations increase, and for quite some time they remain high. Of course, if one follows this through, one will see these concentrations to decrease again, but at a slow rate. As in organismal pharmacokinetics, in ocular pharmacokinetics, we also have some biotransformation reactions taking place. And it is impressive that tissues such as cornea turn out to contain a considerable number of active CYP450 enzymes, isozymes, but also other metabolic or drug biotransformation enzymes, as the ones that you can see in this table in the right-hand side of the slide. And that creates, of course, a potential for drug biotransformation during absorption of topical formulation. Now, the same is also true for transport proteins, which of course are very important in pharmacokinetics. What we see here in the left-hand side of the slide, those gray boxes, is that cornea and the retina are the two ocular tissues that contain most of the transport proteins. And this is to be expected, I guess, because nature has created mechanisms for things to either, good things for the eye, of course, to go into the eye, and for things that can be perceived as hazardous for the ocular tissues to be able to be removed as fast as possible. And this, of course, creates a potential for drug-drug interactions, because if one is administered a drug which serves as a substrate for these efflux proteins, or even something that blocks their effect, okay, this can increase the bioavailability, okay, into the eye. This can be a good thing if the drug has an extensive therapeutic index, but it can be a bad thing if the therapeutic index is narrow. Following all this information, some people have proposed an ocular drug classification system, not unlike the drug classification system that has been proposed for intestinal absorption, whereby drugs are classified as those that have high permeability and low metabolism, which are, by the way, most of the ocular drugs that are administered topically. Drugs that have high permeability and high metabolism, not too many of those, only one in this particular graph. And then again, drugs that have low permeability and low metabolism, and low permeability and high metabolism. And as I said before, most of the clinically useful drugs fall into this category as expected. Now, another way of administering drugs, clinically useful way of putting drugs into the eye is through the intravitreal administration, which can achieve high concentrations and half-lives and slow release towards other compartments. As we can see here, if you compare the maximum concentrations of bevacizumab injected into the vitreous humor, we can see that it goes from 400 to 37.7 to 300.3, okay, from the vitreous body to the aqueous humor to serum. And a similar, quite dramatic decrease is also observed with respect to the area under the curve. So this system also works somehow like a sustained release system. Now, all this can be combined into pharmacokinetic modeling. Today, there's plenty of information available to design in silico pharmacokinetic models with different compartments and constants of movement of drugs from one compartment to the other, and so on and so forth. So there's quite a lot of experience accumulated in this field. Now, something that we should not forget is that overwhelming the ability of the eye to absorb a drug can lead to systemic adverse effects. As I said before, this is easily forgotten, unfortunately, especially if the drug has a narrow therapeutic index. And those adverse effects can be either dose-dependent or allergic in nature. Conjunctival hyperemia can increase adverse effect risk because more drug goes into the systemic circulation through the conjunctival capillaries when that happens. And of course, the risk is higher for pediatric and elderly patients, as we know from the pharmacology of these extremes of age patients, either too small or too high, for a variety of reasons that have, of course, to do with either the metabolic capacity of the liver metabolic capacity, hepatic metabolic capacity, and the renal function. This is not a problem to be taken lightly. As we can see, there are drugs that can create quite serious adverse effects. Timolol is probably the #1 drug in this category. Quite a number of publications in PubMed. And lately, there are some publications that connect the propensity for systemic adverse reactions, adverse effects, to the genotype of some metabolic enzymes. In this particular example, this is the CYP2D6 homozygous 4 allele carrier. Means that this person has essentially no SYP2D6 activity, which means that the levels of timolol tend to increase in this case, because this is an isozyme which is important for timolol metabolism, biotransformation. But as we can see here, there are other drugs that are reported of producing adverse effects, such as atropine, and of course topical corticosteroids. Depending on the duration of exposure and the time that the patient is exposed to the drug. In this particular case report, those adverse effects were produced after a year-long exposure to the drug. So this is not something that happens with acute exposure, at least not in the case of corticosteroids, but in the case of drugs like timolol, one should expect adverse effects to become apparent even with acute exposure. So to conclude this short presentation, the eye and periocular tissue constitute a system with unique pharmacokinetic properties. Their study and careful characterization is important for the design of ocular drugs and for preventing their systemic adverse effects, but also for understanding the propensity for ocular toxicity of certain systemically administered drugs, something that this presentation was not designed to show, because unfortunately we don't have the time for that. But this is another very important chapter in ocular toxicity of drugs. And I guess that will be all. There's some useful literature covering a large time span. As you can see, one of those articles is as old as it was published as old as 1981, but I can assure you this is a real classic. So I recommend this particular publication for those of you who want to delve into ocular pharmacokinetics. And thank you for your attention, of course.
Thank you very, very much, Professor Voulas, for this excellent talk, with presenting the framework within which ocular medications work. They reach the target tissues and they manage to achieve the therapeutic concentrations, and also for hinting on important topics such as toxicity, adverse effects, systemic effects that need to be taken into account when we prescribe or even when we consider a new drug. I thank you very much again, Professor Voulas, and I think it is good time to move forward to our second esteemed presenter, Professor Dimitrios Fatouros. Professor Fatouros, apart from being a good friend from the old times, is also an esteemed member of the Aristotle University of Thessaloniki. If that is the case, I will restrain myself from further positive words about Professor Fatouros, and I will instead keep them all for Dr. Nicolaidou. I'd like to thank Dr. Nicolaidou for presenting her research work in today's webinar. Dr. Nicolaidou is an ophthalmologist in training. She is currently pursuing her PhD in Germany, and she has already, despite her very young age, demonstrated a great production of significant research, holder of 2 MSc titles, and concluding her PhD now in Tübingen in Germany. Anna, you have the floor. I expect you to take us by surprise.
Thank you very much. I'm very happy to be among you. And I'd like to present to you, it's actually my master thesis project that recently got published to the special issue of Pharmaceuticals. And it's a literature review, as you can see, about the use of nanoparticles in treating AMD. So without further ado, I'd like to just briefly introduce the topic and why do we want to see the intersection between ophthalmology and nanomedicine. So these two key areas that this presentation focuses on. So I don't know until when you heard me, but I didn't say much, so don't worry. So I would like to introduce the subject by saying why we want to merge these two exciting fields, ophthalmology and nanomedicine. So in ophthalmology, we face the challenge of visual impairment rising globally, and today it's already 1 billion people suffering from visual impairment, and this is expected also to rise in the future. So it looks like we need to find better ways to treat vision loss, and one of the main reasons for vision loss is AMD. What nanomedicine comes to offer then is actually targeting, so delivering drugs directly to ocular tissues, as Professor Goulas mentioned, and avoiding systemic exposure. Also sustained release by keeping drug levels stable over time and reducing treatment frequency. I will refer to that in a few seconds. And immune privilege, which means that the eye tolerates these local therapies with minimal immune response. So these benefits, keeping these benefits in mind, let's look into the basics of AMD and the gaps that we have in current treatment options. So AMD, as I guess you already know, affects the macula, which is the center of vision and responsible for sharp vision. And it's the leading cause for irreversible vision loss in older adults. Around 200 million people are affected globally, with the prevalence rising with age. Higher risk is in Caucasians, women, and those who have family history. And as life expectancy raises, so does the urgency to treat this disease. So a little bit of the pathophysiology of AMD, which also will be useful to think about how can we use nanoparticles in this disease. So as you can see in the normal retina, in the upper part of the slide, we have all the different cells functioning well and being on the right place. However, what happens in the early AMD is that we have some inflammation, so activated microglia and activated macrophages come to the retina, and then we have the creation of medium drusen. These two, the microglia and the macrophages, will be useful to see how we use nanoparticles for these two. And then when it comes to geographic atrophy, we have photoreceptor loss, and the RPE starts to be atrophic. Then in the final stage, in the neovascular AMD, we have big vessels coming and breaking the membrane, creating choroidal neovascularization. So all of this, these mechanisms include oxidative stress, inflammation, neovascularizations, and all of these are difficult to target without affecting healthy tissue. The traditional treatments that we have when it comes to dry AMD and wet AMD are practically very limited. The most commonly used one is anti-VEGF injections, which require frequent intravitreal injections, which is usually very I would say, tiring for the patients. And then they have limited long-term improvement. So this poses a burden for the patients, but for the healthcare system as well. And that's why we need a new strategy, some like nanoparticle-based therapies that are actually already being explored. So why do we need nanoparticles? We need a paradigm shift in the treatment of AMD, and that we can do through applications of nanoparticles with drug delivery, photodynamic therapy, and immune modulation. So in my thesis, what we did was that we tried to search through various databases such as PubMed, ScienceDirect, and Google Scholar, with no time restrictions. However, I have to mention that this search was conducted until April 2024, so it's already been a year, and probably some research efforts are not included in this presentation. And we tried to include all original studies encompassing nanoparticles in vivo, ex vivo, or in vitro. And this is an image we've created out of all the results that we found. So we found results in vitro, we found studies being done in mice, chicken, monkeys. However, we did not find any clinical trial in humans yet. And let's see a bit more specifically what did we see in the literature. So the different uses of nanoparticles, you can see on the upper side of the slide. The highest in percentage by far was drug delivery, followed by photodynamic therapy and antioxidant use. Also prominent were the immune modulation and the biomimetic nanoparticles. When it comes to what kind of nanoparticles were they actually using, PLGA were the most used ones, followed by polymeric nanoparticles, as well as gold nanoparticles and lipid ones. Then to put some examples about what we found. So we found that many studies used lipid nanoparticles due to their high stability and minimal toxicity. PLGA nanoparticles showed to be biodegradable and had tunable drug release. And nanosyria were mostly used about their antioxidant properties. Then when it comes to quantum dots, there were studies investigating them about their fluorescent properties for imaging and drug delivery. However, this review did not focus on imaging, rather than we only focused on treatment and management of AMD. And then gold nanoparticles were used for their anti-angiogenic properties. So I present here some example studies that I would like to mention. And as you can see, like, different studies explored different aspects of AMD, from macrophage disguised nanoparticles delivering drugs that reduced angiogenesis, and then other studies that had topical AMD treatment, which means like eye drops. They were trying to use nanoparticles in eye drops and see how effective they are, and they actually proved that they were effective enough. However, this was mostly done in ex vivo studies in chicken. And then actually the number of studies was quite impressive. With the criteria that we posed, we found more than 60 different studies. As you can see, they did not only refer to dry or wet AMD. Some were about both. Some did not specify exactly, or and the most of them were about wet AMD and choroidal neovascularization. When it comes to administration methods, we also try to quantify what the studies were proposing. And it's quite impressive to me that even though we have problems with intravitreal injection, with patients. Still, the most used method was intravitreal injections, but some other studies also were trying to use them topically or subconjunctivally. Then some emerging innovation, especially in the latest literature that we saw, was light-activated nanoparticles, which release drugs under a specific light wavelength. For example, Xu et al. Used red light triggered nanoparticles for targeted CNV suppression. Then gene therapy nanoparticles, which were loaded with plasmids, delivering specific genes that were actually acting as VEGF inhibitors. And then biomimetic nanoparticles, which were mimicking, as we saw, either macrophages, and also there were other studies mimicking the Bruch's membrane for better targeting. Then about gold nanoparticles, a lot of studies were done on those. It could actually be a review only about gold nanoparticles, mostly in silico and not in animals, for radiation dose enhancement. However, all these exciting applications do not come without limitations. Safety and toxicity concerns were always mentioned when it comes to risk of bioaccumulation, oxidative stress, and neurotoxicity, as well as scalability. When it comes to nanoparticles, it's difficult to reproduce them consistently at a clinical grade. And then about regulatory hurdles, the approval of FDA and EMA remains complex, and none of these applications has gotten one. And of course, the cost of synthesis can be expensive. These barriers need to be addressed in nanomedicine and can be the future problems, let's say. But in the future, we will not be alone. I am positive that with the help also of artificial intelligence, we will be able to optimize these formulations without having all the hurdles of experimental design and delivery, and finding the best delivery routes. Probably with artificial intelligence, we will be able to exactly propose specific personalized approaches for its patient and its type of AMD, and simulate the nanoparticles interactions with other ocular tissues. Of course, this remains to be proven, but I think the future is bright, and the research already, as I tried to show you, is wide. So I'd like to thank you for your attention. The article is already published, so you are welcome to look into it and come up to me with questions. My email is there. So yeah, thank you so much. And these are my references.
Thank you, Dr. Nicolaidou, very much for this very interesting review of what's going on when it comes to AMD and nanotechnology. This is, as you very well described, a vital issue as more and more octogenarians fill up our waiting rooms in the ophthalmology clinics and are looking for better, easier, safer solutions to their AMD condition. Thank you again very much.
Thank you.
I'd like to move on to Professor Fatouros, and I hope that the difficulty with connection has been overcome. Professor, sorry, say Mr. President, I'd like to congratulate you myself for your recent election, for your most recent feat to be elected as president of the Pharmaceutical Department of Aristotle University of Thessaloniki, and also to express my gratitude for having you, not because you are an old and loyal friend, but also because you're an esteemed professor and scientist with extensive and thorough work in all aspects of pharmacology, some of which I have been honored to understand up to a point. And
Thank you.
I hopefully we've got a lot to learn about glaucoma, which is another huge field when it comes to clinical ophthalmology. The floor is yours.
Thank you very much for the invitation. Both both Tatiana Buric and Dr. Labrogiannis and Professor Chinopoulos. I do apologize. I have some problems with my connections, so I really do apologize for that. So actually, the title of my presentation is Novel Nanomedicines for the Treatment of Glaucoma. So can you see the next slide, please?
Yes.
Can you see the next? Yes, okay, great. So actually, glaucoma is a disease which is associated with limited drainage of the aqueous humor, and it can increase the intraocular pressure. And that could lead to progressive damage to the optical nerve and permanent loss of vision. It affects at worldwide 60 million of people, and the estimation is that by 2040, 110 million people will be affected from glaucoma. So what is the typical treatment? Is topical drug administration in an attempt to decrease the intraocular pressure by enhancing either the aqueous humor drainage or by reducing aqueous humor production. Most ophthalmic formulations are in the form of eye drops. However, major issues have been reported with the eye drops, which are mainly connected with poor drug bioavailability to the target issue. In some cases, it's less than 5%, and actually this is associated with it is actually due to the lacrimal, drainage, and also in some cases we have a rapid tear turnover, and that means that we have short residence times between 1 or 3 minutes. Also, there are other drawbacks like dosing frequency, dosing variation, and also we have reports for blurred vision. So all these things lead to poor patient's compliance. Although there are very limited studies on the formulation of solid dosage forms for the eye, these formulations could have certain advantages over the traditional formulations. We know that solid dosage forms do not contain water. So in this case, we have improved storage stability, and also this could result to prolonged ocular residence. And ultimately, we will have better bioavailability. Also, we could have less systemic side effects, and we have better dosing accuracy with the solid dosage forms, more convenient in the dosing frequency, and all this could lead to a better patient's compliance. So what could be the ideal solid dosage form? A solid dosage form that could be converted to a clear film upon contact with the corneal surface. And actually today I'm going to present you some work we have done in our lab working with solid dosage forms for ocular drug delivery. For our formulation, we worked with electrospinning. Electrospinning is an emerging technology. It belongs to this family with other technologies like microfluidics or 3D printing. And for the people that are not so familiar with this technique, it's actually quite simple. We have a syringe with our polymer solution that contains our API, and in the needle, at the end of the needle, we apply sufficiently high voltage. So the liquid droplet is stretched and erupts from the needle. At that point, we have the production of the Taylor cone, and it's called the Taylor cone because of the scientist, Professor Taylor, that some decades before developed this technique. So in our case, we would like to develop these nanofibrous films that could be easily adapted to the cornea. And due to the advantages of this technique, we can get highly porous structures, and also we have fibers with high surface to volume ratio. They have also, these fibers, the ability to accommodate both hydrophilic and lipophilic compounds. And more importantly, with this technique, we can achieve the drug amorphization. And with the drug amorphization, actually we have better solubility and possibly better absorption to the site of action. So, moving to the next slide, the materials we worked with was polyvinyl alcohol, is a water-soluble synthetic polymer which has got the ability to absorb water, so it has got good swelling properties. Also poloxamer, which is a synthetic non-ionic amphiphilic block copolymer, and also it has both mucoadhesive and thermoresponsive properties. And for the as an API, we use the timolol is a well-known beta blocker, and it is used. It works really nice for the treatment of glaucoma. So briefly, we optimized the production of our formulations. We characterized them. We assessed. We did the in vitro assessment, ex vivo evaluation, and finally we did the in vivo pharmacokinetics. For the optimization of electrospinning conditions, you see in the table we worked with PVA. We varied the amount of poloxamer, and also for the electrospinning, the solutions that should be used for the electrospinning should have certain properties in terms of the surface tension, the viscosity, and the conductivity. So all these studies are necessary before we will start to do our work with electrospinning. As you can see, the conductivity values are presented here in the table and are in the acceptable range for solutions to be used for electrospinning. And also we did the viscosity studies of our solutions, and what we saw is that we have a shear thinning behavior. We reached the Newtonian plateau, and actually what we saw, which is normal, we had increased viscosity when also increased the amount of the polymer to our solution. And to the left part of the slide, you can see the electrospinning parameters. We used water as a solvent, and then we used aluminum foil to collect our electrospan fibers. We moved to the physicochemical characterization, starting with the visualization studies. We performed scanning electron microscopy analysis. As you can see, we received smooth nanofibers. Actually, they make a dense 3D network. Their orientation was random, and the presence of timolol malate actually didn't affect significantly the thickness of the nanofibers. So as you can see, we didn't have big variations in plane and drug-loaded nanofibers. We performed also routine analysis, which is typical when we're working with solid formulations that includes FTIR, X-ray diffraction, differential scanning calorimetry. So briefly, I'm not going to get into details. Briefly, from all these studies, the result we got is that our drug is in an amorphous condition. We can see it now nicely here, where all the peaks disappear when we have the formulation with the poloxamer and the PVP. The same results, and they were in good correlation with The XRD analysis also was with a differential scanning calorimetry. Again, the typical endothermic peak of timolol malate actually disappears when we have the formulation containing both poloxamer and PVA. So the results was that our formulation, our drug was in an amorphous state. Another important issue is the wetting time, because we are working with solid formulations, and actually it's really important. Once we place them in the eyes, we don't have any problems with our vision. So in that case, we did a wetting test, a simple approach. In a petri dish, we had soaked Whatman paper with simulated tear fluids. We added our film, our nanofibers, and actually, as you can see, in most cases, for most of the formulations, the wetting time was between 4 up to 1.4 seconds. Actually, this is acceptable, and it is suitable for ocular applications. And why it can be the wetting time is so short, because we have high porosity and hydrophilicity because of their own materials. Also, we noticed that the formulation that contained the highest amount of poloxamer had the faster wetting time, 1.4 seconds, and this is due to the surfactant properties of poloxamer. We moved and to the urological studies. Briefly, we noticed that the storage modulus for all these formulations was higher compared to the loss modulus, and this is that means that we have a gel-like structure, which favoring the in situ gelling in the eye. Also, we had viscoelastic behavior, and this is really good when we're talking for formulations for ocular drug delivery. We did also the in vitro studies, starting with the drug loading and the encapsulation efficiency. It's actually remarkable that we achieved drug-loaded values closely to the usually administered doses of timolol malate for glaucoma, and that was this amount was loaded in a relatively small quantity of nanofibers, 1.5 up to 4 milligrams, and that means also that in that case we have a small volume of this solid dosage form. Regarding now the in vitro release profiles, this was performed in simulated tear fluids, and as you can see, almost 80% of our formulation was released within 5 minutes, and almost 100% in 15 minutes. Actually, we had first order kinetics, and because we have water-soluble drug in a porous matrices. In most cases, the release profiles were similar. For example, with the exception of the formulation that didn't contain any poloxamer, and that was because it had a slow onset because of the reduced wettability of our formulation. Then we moved to the cytotoxicity studies, and actually there are many ways that we can perform cytotoxicity studies. So one is with using proper cell lines. In our case, we worked with the chorioaladoic membrane. In that case, we use fertile eggs. So in that case, there is a certain procedure with this. It's an ex ovo method actually, and you can get an estimation of irritation potential for formulations for ocular use. You can see the hyperemia, the hemorrhage, and the coagulation. And actually what you do, you apply to the fertile egg in a small opening of the cell your formulations for 5 minutes, and then you measure the irritation potential using a kind of score. So actually what you can see is that figure 1 is the negative control. This is the one, the sample that was just incubated, treated, with sodium chloride with saline buffer. So in this case, we can see that the vascular system is complete intact. Formulation figure B, C, D, and E is our formulation. So you can see again that practically we have no irritation, but when we use sodium hydroxide, this is this one, you can see that our vascular system network was completely damaged. So that was a good indication that our formulation is actually safe for further use. We moved to ex vivo studies. Ex vivo studies are usually performed with French diffusion cells. In our case, we worked with vertical. They have the donor and the acceptor phase, and you can see here in between, you will we put the membrane. In our case, it was porcine corneas, and we performed permeability studies for 4 hours. We measured the steady state flux and the apparent permeability. Actually, we didn't see big differences between the formulations, between the 4 formulations, but in all cases our formulations work better compared with the solution of timolol malate. Moving to histological studies. Again, we did histological studies for the different formulations. You can see in figure A is the control. Very nicely, we can see the epithelium, the Bowman membrane, the stroma, the Descemet membrane, and the endothelium. In all formulations, we had a mild to moderate superficial desquamation. You can see it actually here. We had a small degree of swelling of stroma, and in some cases we had vacuolation of the epithelium. Finally, we did some in vivo studies. In that case, we worked with normotensive New Zealand rabbits. You can see here the formulation when we added it to the eye. We tested our formulations and the solution of timolol malate. More or less, we got equivalent intraocular pressure reduction. However, the timolol solution after 24 hours was back to the baseline. However, we had 2 formulations that contained poloxamer, and we had prolongation in the intraocular pressure reduction. So in simple words, that means that we could have reduced doses. Also, we did the ocular tolerance assay, which is the Drake test. In most cases, the score was less than 3. That means that we have a non-irritating formulation. So, to conclude, the electrospan ocular films were successfully developed, comprised of PVA co-formulated with poloxamer and timolol malate, were fast dissolving. They had viscoelastic behavior, and also they were safe based on the head cam assay. Also, the permeability studies, regarding the permeability studies, our formulation performed better compared to the timolol solution, and also the intraocular pressure and also for the in vivo studies, our formulations were better compared to the Timolol solutions. I would like to thank my colleagues, Dr. Christina Karavasili, who is currently an assistant professor in our department, and Mr. Ioannis Andreadis. He is currently at AstraZeneca. He's working as senior scientist there, and our work was published back in 2022 in Molecular Pharmaceutics. Also, I would like to thank Onassis Foundations and Latsis Foundation for the financial support, and thank you for your patience.
Thank you very much, Professor Fatouros, for this very interesting talk on which addresses the technique of electrospinning, which is there is so much to know about it from a clinical perspective. And as time seems to pass, I would like to pass the torch to the great Ioanna team, initially represented by Dr. Georgios Traiopoulos, a PhD student and an ophthalmologist in training who has done, and this is part of his PhD work that will be presented today. Unfortunately, an unforeseen minor health issue has rendered him unable to present today. Therefore, the captain of the team, Dr. Eleni Bagli, an esteemed ophthalmologist with more than 15 years of experience on related research fields, including stem cells and the ocular barriers, has kindly agreed to help us with the presentation today. Thank you, Dr. Bagli, very much for participating. Thank you, Georgios, for your good work. Hope you get better soon. And I'd like to pass you the torch.
Thank you so much to be with you for the invitation, and I wish Yorgos a quick recovery. I hope you can see my presentation, so I can start. So I'm going to speak about the fabrication of a novel humanized model of the inner blood-brain barrier. I'm going to make a brief introduction. So the neurovascular the retinal neurovascular unit, it's a highly integrated structure that is composed of neural cells, glial cells, and vascular cells, including endothelial cells and pericytes. And it is the interaction between these different cell types which is essential for the retinal homeostasis and also for the coordination of what is called retinal neurovascular coupling. Now, a key functional component of the neurovascular unit is what is called inner blood retinal barrier, which is actually the very tight connection between the endothelial cells and the fact that there is a very controlled selective exchange of substances between the bloodstream and the retinal tissue. Now, we all know that the blood-retinal barrier is very important and it has a crucial role in the onset and progression of most of the retinal diseases, mainly diabetes and central retinal vein occlusion. And we know that most of the times the effect is in the vision of the patients is devastating. And particularly for diabetes, we can say that it can affect all the components of the inner blood-retinal barrier. Now, this makes it clear and highlights the importance that we need to analyze and evaluate the molecular pathways that they govern the physiology as well the dysfunction of the inner blood retinal barrier in order not only to identify new therapeutic targets or even disease biomarkers, but also to design drugs that they could preserve the integrity of the inner blood retinal barrier, or they can cross it selectively, or it can be remained excluded in case we want to avoid any neurotoxicity. Now, if we take a look in the pharma industry, we realize that most of the promising drug candidates fail to progress to clinical trials. And although somebody can find many reasons for that, one main reason is that the preclinical models have very poor translational relevance. If we see them closely, these models, the preclinical models, are standard tissue cell culture models and animal models. Now, regarding the tissue culture assays, we know very well that there are limitations that they have to do with the source of the cells. So, for example, if we want to culture neural cells, it's very difficult to isolate them. And also, as everybody can agree, culturing cells in a 2D system, there is no matter that we can say that it can recapitulate the complicated 3D structure, architecture, and microenvironment of a human organism. Now, on the other side, we have the animal models, and we all know that there are differences between, especially in non-primate animals and humans, that has to do with the structure, the function, and also the genetics. Also, we have ethical concerns, and we have also the cost and the lack of availability many times. As a result, we have a very low prediction value regarding the output that we have in humans, and this we have to see it from the point of view not only of the efficacy of the drug, but also from the point of view of toxicity, because we have to keep in mind that around 30% of the drugs that they are rejected in the clinical trials is because of toxicity issues. Now, specifically for the inner blood-retinal barrier, they exist in preclinical models. Wait. That also they share the limitations that I said before, but we have to keep in mind that they have given us valuable information about the importance of other cell types such as pericytes and astrocytes in order the barrier to be built. But anyways, it is clear now that we need more advanced in vitro systems to better replicate disease mechanisms, identify novel therapeutic targets, and enable drug screening with improved translational relevance and predictive value for human outcomes. Now, during the last 2 decades, there are 2 technologies that they have emerged and they have rapidly advanced in the field. One is the pluripotent stem cell technology and the other is microfluidics. Now, what happens with pluripotent stem cells is that cells terminally differentiated, somatic cells, such as fibroblasts, can be isolated, and then by overexpressing 4 different transcription factors known as Yamanaka factors, named after Yamanaka, who took the Nobel Prize for this, they can be reprogrammed, meaning that they can get into a pluripotent state. They are called induced pluripotent stem cells, and if you apply to them different differentiation protocols, actually they can generate any cell in the organism that belongs to the 3 germ layers, mesoderm, ectoderm, or endoderm. Now, in our lab, we have an experience of at least 15 years working with pluripotent stem cells, reprogramming, genetic manipulation of them, or even differentiation of them to any cell type. The other technology is microfluidics, which actually manipulates very small volumes of fluids in a very precisely engineered microchannel in this kind of system. This is small chips you can see here, and which can mimic very well the scale and the dynamics of a human microvasculature. And it's the fact that we can really control very well this cellular microenvironment that makes the microfluidics an ideal platform for tissue engineering applications. Because by formulating this dynamic fluidics and allowing also the compartmentalized culture of different cell types, we can get very advanced cell systems, very complicated, that they resemble a lot what is the tissue in a human organism, and this is what we call here an organ on a chip. So, based on that, the aim of the project actually is the development is to integrate these 2 cutting-edge technologies, pluripotent stem cell technology and microfluidic system, in order to develop a physiologically relevant in vitro model of inner blood-retinal barrier. Actually, the idea is to differentiate human pluripotent stem cells to the neural and vascular component of the retina, and then assemble that in a microfluidic platform in order to mimic the complicated neurovascular unit interface of humans. Now, the first part of the project is already done in the lab. Actually, it's the generation of the retinal cells from the differentiation of human pluripotent stem cells. And we have established all the protocols that we can generate retina and astrocytes from iPS through ectoderm, and endothelial cells, pericytes, and microglia through mesoderm. I'm not going to speak about this part of the mesoderm populations because we have already published it and very well characterized the cells. So I'm going to focus a little bit on the differentiation of iPS to retina. And why is this? Because retina is a laminar, very complicated, with very complicated structure tissue, and it has at least 7 different cell types of neural cells and glial cells. So in order to be able to generate it in vitro, we follow a stepwise differentiation protocol, and we induce the differentiation of iPS to retina by mimicking the spatiotemporal signaling events that take place during the retinal embryogenesis. So in that way, we succeed finally to have these 3D self-organized 3D structures that they have the laminar architecture and the cell diversity that the retina has. This is a very laborious method that we get the mature photoreceptors in the retinal organoids at least after 3 months of differentiation. And what we get is this is called retinal organoid. Here we see the illustration of it. If you cut it, you see very well, you can see the laminar structure, the architecture, which mimics the one of the retina. We see that the outer segments of the photoreceptors face outwards, while the ganglion cells face inwards. And then you see here underneath, we have a retinal organoid acid. You can see it with in an electron microscope. And also we have fully characterized them here using immunofluorescence and antibodies against particular markers which are characteristic of each cell type in order to confirm that we have developed all the cell types and they have a correct distribution in the retinal organoid. Now, the next part is and this is what we're working on now, is to build our model. Actually, we are in collaboration with Professor Periklis Papadopoulos from the Department of Physics in order to make this microfluidic platform. So actually, we engineer actually a small a micro channel that the flow will be applied. This channel is going to be lined with endothelial cells, and on the top compartment, which is separated with a semi-permeable membrane, we're going to add the rest of the retinal cells like pericytes, microglia, and also the retinal organoid will be cut in order to have the correct orientation. We're going to apply flow and we're going to culture them in order, hopefully, to get what is this structure here, this interface, which is the inner blood retinal barrier. This is going to be further characterized and extensively using live cell imaging confocal microscopy to confirm that we have the tight junctions, the tight junction proteins, that they have a correct distribution in the cell using immunostaining and immunofluorescence. We're going also to perform functional assays like paracellular permeability assays or transcytosis assays to confirm that we have a very good barrier. And then we are going to finally apply pathological conditions, actually diabetic conditions, in order to see that we can get changes which are pathophysiological changes that are characteristic of the diabetic retinopathy. So as a conclusion, we hopefully have shown this humanized inner blood retinal barrier model that we can use it to study physiology and pathophysiology of inner blood retinal barrier, to recognize like that new therapeutic target, mainly for diabetes, but also and mainly to use it as a screening platform for drugs, for drug toxicity, and also for gene therapy vectors. So this is the group. We are in the Institute of Biomedical Research that is in Ioannina. And thank you so much for your attention.
Thank you very much, Dr. Bakli, for this very interesting talk. Thank you, Yorgos, for your hard work as a part of this, as a valuable part of this team. And hopefully your model will find a number of applications on it, not only to study the barriers, as you said, in itself, but also in preparing new formulations and new means of drug delivery. I'd like to take this opportunity to ask our participants if they have any questions. I see one question in the Q&A box by Dr. Zan Andari, if I'm not mispronouncing, and it is addressed to Professor Antonios Goulas. And Dr. Andari is asking, Professor, what is the influence of the tonicity on the ocular drug bioavailability?
Well, tonicity does have an effect also, of course. But I think Professor Fatouros is probably more of an expert to answer this question.
I guess, at least in our case, we're working with isotonic formulations so far for ocular drug delivery systems. But yeah.
Thank you, Professor Fatouros. And if I may ask a couple of questions myself, that's okay, Tatiana, if we've got a couple more minutes. We have exceeded the time just by a tiny bit, so I think I might ask a couple of questions. I'd like to ask Dr. Nicolaidou, with your experience so far, how do you see artificial intelligence being integrated in the treatment of AMD using nanotechnology? What is the current status from your research so far? What do you think?
So the thing is, it's not that artificial intelligence will help in the treatment of AMD. I mean, probably yes, but in other ways, and not through nanomedicine. What I've seen during the literature search was mostly the fact that with AI now we have the privilege of, for example, instead of testing all the different types of nanoparticles, we can predict which nanoparticle will be better suited for which kind of application. So this prediction actually saves us a lot of money and a lot of years of research. So if this prediction is accurate, then we can guide the therapeutic approach better and have faster clinical applications. Then, of course, this has also risks depending on what database is the algorithm based on, and if there are bias on these databases. So it needs to be a really careful integration of AI, but in general, it seems that it will help in the manufacturing most prominently. Yeah, and I think we have we don't have this in my review, but we have seen another review published about AI and nanoparticles manufacturing. So yeah, there are also reviews on that, but still it's really this field of research has just opened and there's a lot of expectations, and of course this comes always with questions. So yeah, I mean.
So there's a lot more to come in the following years. Is that right?
Yes,
You expect to
definitely.
see more and more of these combined.
Yes, and especially if you look at the in silico studies, which is like modeling studies already for the gold nanoparticles, then like we're already working with theoretical models, not real tissues. There are already studies on that. So yeah, it's supposed to be more computated, but then how this will actually apply to clinic, of course, if you do something computationally and then you put it into clinic, a lot of things might be unpredictable. So we need to try and see.
Thank you so much. And it's okay if I ask a brief question to Dr. Baglia as well.
Of course.
Dr. Baglia, judging by your experience both on the research and the clinical level, which conditions would you see as better candidates? Which groups of conditions do you think would benefit more from your research work?
Well, actually, diabetes is because of the great percentage of people suffering from diabetes and the devastating effect that it has in the vision. We all know the diabetic macular edema. Also, the vascular diseases, most of the retinal disease, actually, at one stage of their development, they will have problem with the barrier. So I think this is very important to be solved somehow. And it's true that we face it from only one part of the component. We only see that from the vascular point of view, but I think that the glial cells are very important there. And maybe new therapeutic targets have to do with improving the health of glia and the neural cells, which might be affected far before the vascular component. So let's see.
So I think we've got, between us, we've got plans for glaucoma, AMD, diabetic retinopathy. I think that we are looking forward to putting half of our colleague ophthalmologists out of work if it all works. Let's hope that this doesn't happen, at least in this particular manner. Professor Tsinopoulos has also had problems with his connection, so if it's okay, Tatiana, I will be doing the signing off. I'd like to thank you all for joining us today.
Well, before we close, let me add to the question that was asked before by our viewer. What is important here is to avoid, essentially, to avoid increased tonicity. So, because a hypertonic solution will tend to increase the tear flow. And as we said before, increased tear flow will tend to increase the nasolacrimal drainage and removal of the drug from the outer surface. So yeah, so a smaller tonicity would not be that much of a problem, but a hypertonic solution, yeah, it's probably something that you don't want.
Thank you very much, Professor Willis, for this valuable insight. And I'd like to thank you all for joining in today and for providing some aspects of your great work in your fields. It's an honor to have you all in this webinar. And if we don't need to add anything, Ms. Beretz, could we conclude here and thank our devoted viewers for their attendance?
Yes, definitely. Thank you all for joining. Of course, our chairs and our speakers for their hard work. We didn't have luck on our side today with technical issues, but it was definitely worth it, all of our time. So thank you all for attending and for joining us today.
Thank you very much.
Thank you for inviting us also, and I would like to also congratulate the speakers for some excellent basic research that we witnessed today.
Thank you all very much. Have a nice evening.
Thank you.
Well, thank you. I just wrote in the chat box how to get the certificates for attendees, and that is it. We conclude for tonight. So have a great evening, and we will see you the next time.
Thank you so much. Thank you. Have a nice evening.
Bye, everyone.